Every single complex three-dimensional human body actually begins its early development as a completely flat two-dimensional plate of cells. This specific topographical baseline is reached at the end of the third week of gestation. At this stage, the developing human is a trilaminar embryonic disc, a flat, three-layered structure that serves as the starting parameter for the folding process. We can see the strict spatial arrangement of these layers here. The ectoderm forms the dorsal layer, highlighted in blue, with the pink mesoderm in the middle and the yellow endoderm forming the ventral surface. The amniotic cavity sits directly above that blue ectoderm. Positioned directly below the yellow endoderm is the yolk sac, also called the umbilical vesicle. Running along the dorsal midline of the ectoderm is the neural plate, the precursor to the central nervous system. To ultimately establish the definitive human body plan, this flat embryonic geometry must transition into a closed, multi-layered cylinder. Embryonic folding occurs as a forced mechanical reaction to constrained space. This top-down view shows the physical catalyst, the explosive growth of the neural tube along the midline, compared to the slower growth of the embryonic margins. Because the outer edges cannot expand fast enough to accommodate the lengthening center, the disc buckles downward. This is known as differential growth, and it is the physical mechanism behind the cephalo-caudal transformation. This growth triggers two simultaneous longitudinal folds, the cephalic fold at the head and the caudal fold at the tail. That explosive expansion of neural tissue acts as an internal engine, physically dragging the flat margins ventrally to construct the three-dimensional torso. This diagram illustrates the embryo's cranial half. The flat alignment of the blue neural tube, heart tube, and septum transversum establishes a baseline. In panel B, the neural tissue swells as forebrain vesicles expand. By panel C, the developing forebrain severely overhangs ventrally. This creates a direct mechanical consequence for the yellow endodermal layer located directly beneath it. The overhanging fold pinches the yolk sac, incorporating a portion of it directly into the newly forming head region. This incorporated blind-ending endodermal tube is the foregut. The forward thrust of the developing brain is the physical force that manufactures the upper digestive and respiratory tubes. To understand the resulting shift, we have to look at the pre-folding positions of the cardiogenic area and the septum transversum. Initially, the primitive heart begins its development completely outside and above the primitive brain. As the brain folds forward, the red heart tube and surrounding mesoderm rotate 180 degrees, swinging backwards beneath the folding head. This establishes the definitive location of the heart tube on the ventral surface of the embryo, inferior to the foregut. The septum transversum is simultaneously relocated to a position just caudal to the heart. This septum will eventually form the central tendon of the diaphragm, creating the physical division between the chest and the abdomen. Without the mechanical leverage of the head fold, the heart would remain outside the cranial vault rather than secured inside the thoracic cavity. At the opposite end of the disc, the same process is occurring simultaneously. This diagram of the caudal eminence maps the pre-fold anatomy. Panel A shows the flat relationship between the primitive streak, cloacal membrane, and connecting stalk. In panel B, the distal blue neural tube elongates, pushing the primitive streak completely caudal to the cloacal membrane. This tail fold also captures the posterior portion of the yolk sac endoderm. This newly internal tube is the hindgut. The terminal end of this hindgut dilates slightly to form the cloaca. As the fold completes, the connecting stalk and the allantois are dragged onto the ventral surface of the embryo. The tail fold uses the same mechanism of neural expansion to pull the future abdominal and pelvic anchors into their definitive ventral alignment. While the head and tail are folding longitudinally, the lateral body walls are simultaneously folding inward from the sides. These four folding margins, cranial, caudal, left, and right, all converge at the embryo's ventral center. Their meeting point constricts the connection to the yolk sac into a narrow, localized tube. This results in a continuous internal Beck tube. The restricted connection seen here is the omphaloenteric duct, or vitelline duct, which maintains the only remaining link to the yolk sac. Simultaneously, the blue amniotic membrane expands to pull ventrally and encase the entire embryo. This expansion bundles the connecting stalk and omphaloenteric duct together, forming the epithelial covering of the umbilical cord. By this stage, the final topography is established: a dorsal neural tube, a fully internalized and segmented gut tube, and a ventrally secured heart. By the end of week 4, the mechanical folding process has successfully transformed the two-dimensional disc into a three-dimensional tubular organism ready for organogenesis.